All right. Okay. So second to last question we need to talk about is this one. Why are dipterans some of the most dangerous animals on Earth? Why are dipterans some of the most dangerous animals on Earth? Because this is an interesting question. I don't know if you know what dipterans are or not, but they are flies, mosquitoes, gnats. What else is in there? That's a pretty good. Oh, no, ticks aren't in there. It's a single order of insects. Because when I think of dangerous animals, I tend to think of, I don't know, a thing like a hippopotamus, which kills more people than any other animal in Africa every year. And just crazy, right? Crazy dangerous. But the number of people, I don't know if you knew that or not, but hippopotamuses, they are angry. They are angry animals. And they don't even eat. I mean, they eat plants. They eat a lot, but they eat plants. Like, they kill people not to eat them just because they hate them, right? Just because they're angry. I usually don't think of flies and mosquitoes when I think about dangerous animals, but there's no doubt that this order of insects results in more human deaths every year than any other group of animals, than all other group of animals combined, tenfold. I mean, it's crazy. And one of the main reasons for that is mosquitoes. A number of different species of mosquitoes are intermediate hosts for Plasmodium species. So Plasmodium species are the causative agent of malaria. And so malaria alone accounts for approximately 20 million deaths each year, although that number is probably underrepresentative of the true number. So malaria tends to be most dangerous in areas of the world where we don't have great health care, we don't have great records, so it's probably a smaller number than the actual number. And so that's one of the main reasons. We'll talk a little bit more about Plasmodium in a moment. Plasmodium is the protist, what this chapter is all about. Mosquitoes are not the protist. Okay? So the reason why dipterans are so dangerous is because of the protists that they carry from host to host. Tsetse flies, another type of dipteran, transmits Trypanosoma brucei gambiense, which is the causative agent of African sleeping sickness. Does it result in as many deaths as malaria does, but still results in a number of deaths every year and in reduced vitality. It's an incredibly difficult illness to deal with. So again, the reason why dipterans are so dangerous is not because, you know, when they land on your nachos, you don't want to eat them anymore, right? I have a story about that. If you want to hear it, we can talk about that later. It was not me. I don't care if dipterans land in my nachos. I will eat them. But anyways, it's another story for another time. But it's not because of pathology that they are creating. It's because of the protists that they carry from host to host. And so right out of the textbook, you have some life cycles. These are really, really nice life cycles. And what I mean by really, really nice life cycles is these are just great sources of, I don't know, test questions, just, you know, wonderful opportunities to talk about as we talk about protists, because they're parasites. It's just wonderful. I mean, it's actually not wonderful, it's horrific, but it gives you a lot of interesting things to talk about. So these parasites actually require both a human host and an insect host in order to complete their life cycle. They cannot complete their life cycle without both hosts. So it's not like this is just something that we are accidentally getting by being bitten by tsetse flies. It's this, the parasite is manipulating tsetse flies for their growth inside the tsetse fly and then to transmit them to humans that they also need to complete their life cycle. And so what you find as you look at life cycles like this is you find reference to infective stage, and the key there is, so we'll see this in step 5 right here. Trypanosoma brucei transforms into infective stage. And so whenever you see that in a life cycle, it is an indication that there is development happening within that host. Which means if there is development happening within that host, there's some pretty significant life events happening for that parasite inside of that host. Yeah, Mika. Oh yeah. Yeah, tsetse flies are parasites themselves, and they feed on blood. They require blood meals. The females require a hefty blood meal in order to make eggs, in order to reproduce. Yeah.
So is a regular housefly a parasite?
No. Although some people will put them into that category because of their potential to cause pathology, but no. Okay. No. And so you also see in the life cycle, it tends to reference where in the host you find this parasite. And so here you find it in the blood, lymph, and spinal fluid, basically in many different fluids inside of the organism. All right. So again, both hosts required for that parasite to develop properly. That's an important idea that we're going to come back to when we get into animals and we start talking about animal parasites, because it's a common theme of parasites, that they have very complex life cycles and require multiple hosts in order to carry out their life cycle, in order to reach sexual maturity, make offspring. And it's just strange. Like, how did that come about? How did that come about? It's a wonderful question. So here is the life cycle for Plasmodium. There are a number of species of Plasmodium that cause malaria. There's Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, Plasmodium ovale. There's several different species, and I just tell you that not because you need to know all those species, but I just want you to know that on top of having an incredibly complex life cycle, which how many rings do you see in this life cycle? 3, right? And the last one, how many did we have? We had 2. We had a human host, and we had a tsetse fly host. Here we have 3, and you have 2 different tissues within the human host. You have a cycle in the liver, and you have a cycle in red blood cells. And so this parasite actually has 3 hosts. 2 of them just happen to be in the same organism, but 2 different tissue types in the same organism. Incredibly complex life cycle. Okay? 3 different hosts, all required for the development of this parasite to reach sexual maturity, to get through all of its developmental stages. Yeah, Emerson.
If it's so complex, why do they do so well?
That's a wonderful question, because you're like, wow, this just seems way more difficult than it should be, but mosquitoes are voracious feeders, right? And so mosquitoes are more than happy to feed on the other host of this parasite, and the mosquitoes don't need to be thinking like, hey, I'm doing this to serve my Plasmodium. They're really just doing it so that they can get that blood meal and make their eggs. But because of how voracious mosquitoes feed, or how voracious of feeders that they are, this happens all the time. So even though it's really complicated, it's reliable, right? It's really reliable. Yeah?
When you say that it takes multiple hosts to get through a life cycle, does that mean that they won't die until they've reached those?
Not necessarily. It means that they won't complete their life cycle. Just like, I mean, a human could die before they reach sexual maturity, right?
The life cycle means that they've made more of
They've reached sexual maturity. They've reached the stage that they could, and so they could complete their life cycle and still never make offspring. It's just they've completed their life cycle. They've reached sexual maturity and got to a point where they could make copies of themselves. Because remember, we consider that a characteristic of life or a characteristic of living forms is to reproduce, is to make copies of themselves. So if they don't reach sexual maturity, they have not in fact completed the life cycle. They're missing one of the characteristics of life. Okay? So this is an incredibly complex life cycle. 3 hosts, essentially. 2 of them just happen to be in the same organism, liver cells and then red blood cells. And then on top of that, there's a wide variety of different species, all in the same genus, that have a very similar life cycle. And so now we don't just have one parasite with this really complex life cycle, but we have several parasites. And then if you count other forms of malaria that don't infect humans, like avian malaria, other types of malaria that only affect other types of mammals, we've got dozens, if not more, different species of parasites with very similar life cycles. And this is strange. Like, how do these come to exist? How do these come to exist? Okay? And so what to take home from this slide and the previous is that if you're given a life cycle like this, let's just say hypothetically a week from today, you were doing some kind of an assessment, okay? Hypothetically. And you were given one of these pictures, and it just asked you a question of like, hey, explain some of the implications of this life cycle. You might be expected to have to go through and say, okay, well, we've got 3 rings here, which is indicating that we have 3 different hosts required for this protist to carry out its life cycle. That's an implication of this life history or this life cycle diagram. Another implication is that, I mean, you from here you can get, you know, like very focused and say, okay, well, we can just go like point by point, right? It's walking you through this life cycle, stage 1, stage 2, stage 3, stage 4, stage 5, stage 6, stage 7, 8, 9, 10, right? So other implications is that in order for, I don't know, the organism to produce gametes, it has to leave this section and move into its red blood cell portion of its life cycle, okay? So you can get very, very specific with it by information provided right in the life cycle diagram. Right? Or you could get, I don't know, a little bit more interesting with your description, use your powers of critical thinking, and to say something like, the really successful individuals of this parasite are probably going to do a good job at getting from one host to another. So particularly successful individuals of this parasite are going to be particularly good at getting from one host to another, which, if you want to get from a mosquito to a human, you need that mosquito to survive long enough to feed on the human that you left to go into that mosquito, and then to feed on another human so that your offspring have an opportunity to complete their life cycle, right? It makes sense? Okay, so that's like critical thinking. That's not something I expect you to be able to communicate about. I'm just saying that's an option at your disposal, right? Is you can use information from this life cycle to do some critical thinking activities. Allison and then Autumn.
So if, like, malaria has to go through all these different steps, how does the infection grow in the human body?
It's a wonderful question. So most of the pathology is associated with this portion of the life cycle here, the portion in red blood cells, because they're infecting red blood cells. And as they do, they destroy these red blood cells. And so it's showing you the parasite here, although here, oh, there it is right there. You see that? These are reproducing inside of a red blood cell, and they blew that thing up. Okay? That's what most of the pathology is a factor of. This combined with your immune system reacting to their presence. Every time they show up in the bloodstream, your immune system reacts to the presence. You get a fever. And so since then they go and hide out in red blood cells again, something that's characteristic of malaria are what are called paroxysms. This is additional information, but like every 72 hours you get a spike in your fever. Or with some species it's every 48 hours, because it's when these parasites leave the red blood cell, your immune system sees them again, reacts to their presence, you get the fever and the chills and characteristics of your immune system really trying hard to attack something that's there. But that's where most of the pathology is destroying red blood cells. Yeah, and then your immune system reacting to their presence. Does that answer your question?
So it's continuing to multiply.
Yes. Yeah, and it'll just keep multiplying, because you've got I mean, and until you've exhausted all of the individuals that came in the initial bite, but that's not very likely to happen. Okay, there are tons. Plus, they'll stick around in the liver stage for a long time sometimes, and then they'll just, you know, over years emerge and move into this portion of the life cycle. And in a single bite, I mean, you could get hundreds of millions of individual parasites. So, and then, yeah, they actually, what's fun is there's some of these terms we haven't talked about, and we won't, but some of these terms are indicative of how they actually reproduce during this stage. In this stage, this is asexual reproduction, but one individual can produce thousands of the next stage, or tens of thousands of the next stage. So it can multiply very quickly, even though they're not sexually reproducing yet. They're reproducing asexually. Yeah. Autumn.
People who have had malaria can't give blood, right?
If you no, you can't. There's a certain time allotted to that. Now they usually do it a year, like let's say the Red Cross. For a year after you go to a place where when malaria is endemic, you can't donate blood. If you've actually tested positive for malaria, you could still donate blood. You would just have to demonstrate that you no longer have malaria. But again, there are lots of avenues to donate blood. Would the Red Cross ever let you donate blood again if you had malaria? Probably, but there's a time issue involved. Yeah, Emerson.
Because I didn't realize, but malaria is super treatable because when I lived in New Zealand,
Some species are super treatable. Yeah.
Where do they? So there's like
an anti-parasitic.
Bacteria. Where does that stop it?
Oh,
Yeah.
No, that's a good question. So there are two different ways to treat malaria. One, you can do it prophylactically, and so what you do is you basically give almost the equivalent of a vaccine. Right, you give your immune system something to recognize, and then so as soon as they would either end up in the blood from a bite, or as they emerge from the liver, your immune system overwhelms it. Another thing that you can do is target specific stages in the life of this organism, and knowing that most of the pathology is here, you could target specifically these life stages at the emergence. Yeah, Jack.
At what point do you start to like when you get bit by a mosquito,
Oh yeah, like what's
is that when it's in the blood? And
yeah, so that's right here. So step one is the mosquito takes a blood meal and actually deposits, larval forms of this parasite or immature forms of this parasite. This stage, yeah, there's so you call oh gosh, the term is inoculation time, or what's that? Incubation time. It can vary a lot in malaria because this stage could literally last years. And there are some species that will hide away in your liver cells for decades. And then something will happen and they'll emerge and you'll get malaria 40 years after you were in a place where you actually got malaria. Hayden.
How about people with sickle cell? How would the parasite be able to thrive in it?
Yeah, it doesn't. And that's why if you've got sickle cell anemia, your red blood cells have a very hard time, or let's say it this way, the parasite has a very hard time infecting those red blood cells. And so people with sickle cell anemia usually do not get malaria. There are some cases, but they usually do not. And that's the idea of why sickle cell anemia is still around and is still highly prevalent. And you tend to find it highest in places where malaria is endemic. Yeah, Emily.
So the parasite itself is the protist, not...
Yes.
So
although the mosquito is also a parasite, because it's a parasite of humans, and then the protist is a parasite of both humans and the mosquito.
So is that why their life cycles are linked together?
That's what facilitates their life cycles being linked together. The question of why their life cycles is linked together, that's a different question altogether.
So
that enables it to work. But the question of why it's that way actually forces you to ask some other questions that we'll deal with when we talk about animal parasites, of the origins of parasitism just in general. Why are there organisms that feed this way? Period. Regardless of what your view of origins is. Yeah.
So the life cycle of the protist includes the mosquito?
Yes. Yes. Yep. Includes the mosquito. Yeah, and the mosquito is actually where the sexual reproduction happens. Is inside of the mosquito. So the mosquito is what you would call the definitive host. Whatever host the parasite reaches sexual maturity in, that's the definitive host. So, in case In case you were wondering. Yeah, Aiden and then Cameron.
Does each person have to be infected, or can the infection go from human to human?
That's a good question. The only way it can go from human to human is with a significant amount of blood transferring individuals. Or if you got a liver transplant, right? If you got a liver transplant and those liver cells were infected with that. But it would require, I mean, it's more than just like a small amount of fluid transfer. It would be very significant. Like if you were to get a blood transfusion from somebody with malaria, you would get malaria. But if you were to, you know, have sexual intercourse with somebody with malaria, it'd be very unlikely that you would get malaria. Cameron.
In the case that you had, like, for a parasite like this, and then it caused a fever, so usually when we have fevers, we put, like, cool cloths or, like, ice on our head. Does that help keep the fever down or anything?
Oh, sure, yeah. Yeah, I mean, the way you treat a fever would be the same regardless of the. This is just like a cyclical fever. Like every 72 hours or every 48 hours, you get a fever. That's very characteristic of malaria. Yeah. And so here's a zoom-in image of red blood cells infected with one of the species, Plasmodium falciparum, which happens to be the most dangerous of all of the malarial species in terms of its tendency to kill red blood cells. And then here's Trypanosoma brucei, causative agent of African sleeping sickness. Look at these things. What are they hanging out with? Red blood cells. So their pathology is also going to be as a result of their feeding. They're too big to live inside, but they're going to feed as well. All right? Questions?
Those eat red blood cells?
Oh, yeah. I mean, they feed on them. They don't, I mean, yeah.
When we saw the blood cells that were messed up in lab.
Oh, yeah. So that was a different species of Trypanosoma, and they were inside of red blood cells. Yeah.
So they go inside and they eat them from the inside.
Yeah, so we had Trypanosoma cruzi in lab. Is that right? Trypanosoma cruzi. I don't think, we may have had some slides of brucei as well, but cruzi is the causative agent of Chagas disease. Anyways, it's another story for another time. Right? I mean, we can tell tons of stories about pathogens and the diseases that they cause. But those are sad stories. So we're going to keep it happy. This wasn't happy. We're going to move to something happy. All right? Cool. Last question that we have to deal with. How would eukaryotes have developed from prokaryotic cells? How would eukaryotes have developed from prokaryotic cells? And so before I give you this information, we're going to take a lecture break. I know Fridays are a little interesting because you're like, Dr. Engel, like we spend the first 8 minutes doing a quiz. Do we really need a lecture break? The answer is yes. Yes, you do, whether you know it or not. Okay? So how would eukaryotes have developed from prokaryotic cells? What I would like for you to do, we started this discussion last week, and we're not going to finish it today. We're going to continue this discussion today, and I want you to address this question for a couple of minutes with those around you. How would eukaryotes have developed from prokaryotic cells? How would that happen? How would that happen? What do you need? And then how do you get what you need, right? 2 minutes, starting now.
Yeah, but he's talking about one common ancestor. Is it from one common ancestor? Are we looking at?
Oh, that is a wonderful question.
Or is it?
Because remember, like, it doesn't have a context, so it could have had like the photosynthetic prokaryote.
Because it was supposed to believe that one cell.
Or ate a bacteria that produced and then that could have been like the photosynthetic.
So is it
So they would need to somehow gain membrane-bound organelles. Like they would need a lot more organelles than yeah, you need a lot of I think bacteria and archaea, they don't have like a lot of the same organelles that eukaryotes have. So they need activity.
Yeah,
figure out how to do it.
Like the opposite.
So it makes sense that like past
30 seconds.
Or do
All right. All right. Here's some fun information. I'm going to share this with you before we actually talk about what you've come up with. So here are features that you find in all eukaryotes. Okay? A nuclear envelope, mitochondria unless you don't. That's fun. It's like, I thought this is in all eukaryotes. This is why it's so much fun. The cytoskeleton, flagella and or cilia, unless you don't. Chromosomes, that one's always. Mitosis, sexual reproduction, unless you don't. And so this is fun, right? All right, so how do we do it? How do we get this? How do we get a eukaryote from a prokaryotic cell? I had a question up here, and I want to just share it with all of you. And Tara, I saw your hand, but the question is, did it only happen once? Right, this is a fun question. Tara.
By how would you mean, like, what would have needed to take place, or like how would they have changed from prokaryote?
Both.
Okay, so I think there's a lot of like theories about how mitochondria might have been a separate organism before they were combined into eukaryote.
Yeah.
It could have just been the combining of different prokaryotic cells until they formed an organism.
Sure, and that's one of the major ideas. That's probably the dominant idea. Bless you. Is that you have the combination of a couple of different prokaryotic organisms to generate the first eukaryotic organism. That's fun. What else? Any other ideas? Come on, I know y'all had some thoughts. Bless you. Because it wasn't completely silent in here, so unless you were talking about just something random, which is a good probability of that. Yeah, Mika.
It was a bit counterintuitive that there was a hypothesis that prokaryotes devolved from eukaryotes.
Yeah, so there's actually an alternative idea that the original life form was actually a eukaryotic organism and that prokaryotes came from them. It's not a wildly popular idea, but it is something that has been proposed to overcome some of the issues of developing a eukaryotic cell from a prokaryotic cell. Yeah, Allison.
One thing that the text would explain is that some people think the membrane just folded inward and sort of surrounded different components inside.
Including the chromosomes.
Right.
Right, yeah.
That became the nucleus or the different organelles.
Yeah, I mean, and chemically, the membrane surrounding the nucleus is very similar to the membrane surrounding the cell, with the exception of it has different proteins mixed in. But as far as what makes up the bulk of the membrane, they're virtually identical chemically. So much so that that's how you build up the membrane on the outside after you've lost some of it because you pinched it in. Remember we saw phagocytosis, and some things are so big that you actually have to pinch off part of your membrane and draw it in. Well, now you've lost part of your cell membrane, and so you've got to restore it. What you do is you just take some membrane from inside the cell and shuttle it out to fuse with the plasma membrane, because the chemistries are the same. Yeah. Any other thoughts? So yeah, we'd have to get our membrane-bound organelles, including the nucleus, and so an idea is that it folded inward, which is nice. It's a really nice idea. It's a really nice idea. Be better if you actually saw it happen, right? If you saw it happen in some prokaryotic lines that they developed nuclei. But it is a nice idea. That's all. That's all we have. Well, all right, I'll give you some more information. So eukaryotic cells actually in many ways look like the composite of one prokaryotic cell inside of another, what may have at one time been prokaryotic. Because a number of eukaryotic cells have mitochondria or chloroplasts, And those are interesting structures, and we'll talk about why. You have a group, one of our phyla of bacteria that I gave you in that table, right, last Friday. The thing that you remember so fondly because you're like, wow, this is such a wonderful opportunity for me to demonstrate my ability to memorize things. Alpha proteobacteria, one of those phyla, they use aerobic respiration, which means that they use oxygen to generate more energy from what they're feeding on. This is kind of cool. The mitochondrion, we call it the powerhouse of the cell. Remember we talked about that on Wednesday? And it also uses oxygen to generate more energy from food molecules. And so the idea here is that the mitochondrion singular, is actually a descendant of an alpha proteobacterium. And what's kind of cool is if you're thinking of, like, you've got this bacterium, right? And it has its own membrane. And it gets phagocytized by another bacterium. So now you're going to have the original membrane from the bacterium that got eaten, and it's going to be surrounded by the membrane of the new bacterium, right? The one that ate it. And when you look at mitochondria, how many membranes do you think they have around them? They have 2. And it's like, oh man, this picture is so perfect, right? And the inner membrane is really highly folded. And that's how alpha proteobacteria membranes are, because they use aerobic respiration. They pack a bunch of machinery into that membrane, so they need a lot of surface area. And you're like, wow, we've nailed it. We know exactly where mitochondria came from. Yeah, Levi. That's a wonderful question. I mean, mitochondria do replicate on their own. They replicate separate from the cell, but under the control of the cell. It's like, I was going to say it's like my relationship with my children, but I have no control. Yeah. They replicate on their own. I mean, it's crazy. It's like neighborhood kids come over. It's like, I thought I only have 3 kids, and now there are 17 kids in my pantry. Like, I don't even know what happened. So I was going to use that as an analogy, but it wasn't a good one. But yeah, mitochondria, they self-replicate separate from the cell, but the cell controls when and when they don't. The real question isn't how does the cell replicate the mitochondria? The real question is, if that were the case, how did they develop a means of communication? Right? How did the cell know not to eat what it phagocytized for the purpose of eating? Right? What's the mechanism by which you don't eat it? And then how do they start to communicate? Oh yeah, I already mentioned this. The inner membrane has complex folds. We call these cristae, like alpha proteobacteria membranes. Oh yeah, I just mentioned this. Mitochondria divide independently of the cells. They contain their own DNA. They contain their own ribosomes. And it's like, wow, it just looks so cool. Except you still have to come up with how did they develop that communication. And what's fun is they do have their own DNA, but they don't make all of the proteins necessary for the mitochondria to function. Some of those proteins come from DNA made in the cell. So now not only do you need a way to communicate, but you need a way to actually get DNA out of that mitochondrion or bacterium and into the genome of the other bacterium. You're like, well, that's kind of cool. The only thing we've seen so far that does that are viruses. But what if when it ate the alpha proteobacterium, it also ate the phages that infect that, right? Now you have a mechanism by which you can transfer genetic material, right? Because viruses do that. We talked about that. It's wonderful. This is so cool, right? It's so cool. The biggest problem, honestly, and this is one you should write down and you should know, is the language of how you translate mitochondrial genes into protein is completely different than how you translate nuclear genes into protein. The language is completely different. And Most of you have probably learned this, but if you're going to translate from a nucleic acid into a protein, you read 3 bases at a time and you translate it into a specific amino acid. You read 3 bases at a time, so the bases are G, C, A, and T, right? Unless it's RNA, then it's G, C, A, and U, and you read 3 of those at a time and it translates into a specific amino acid. That language, that translation, completely different in the mitochondrion than it is in the nucleus. Emma.
Oh, I was just gonna ask if you could repeat the first thing you said, like the language and
Yeah, the language is completely different. So or another thing that you can use is the code that you use to translate is a completely different code between the mitochondrion compared with the nucleus. So we need to develop a mechanism for them to communicate. We need to come up with a mechanism for them to transfer genetic material. And the biggest problem is we need to come up with a way to actually change the entire language. That's good, right? It's no small task. No small task. Then we have a question, well, are plastids this way? Like chloroplast and other things that not all eukaryotes have, but some eukaryotes have. And the idea here is yes. Yes, it is that way, because chloroplasts have at least 2 membranes around them as well. Some have 3, some have 4. And so the idea is that they actually originated the same way mitochondria did. And so you have a couple of figures in the text, 23.5 and 23.6, that go through and illustrate these events. And these events are known as endosymbiosis. Endo meaning what? Inside. Symbiosis, sim means together. Biosis is life. So life lived together inside, right? Is the whole idea, of endosymbiosis. Okay? So, if you could develop a means by which they could communicate, and you could develop a means by which they can transfer genetic information, and you could develop a means by which the language can completely change, and they can still communicate. Because it's like, the only way you can communicate is if you learn the same language. But they're communicating with different languages. It's really weird. We still don't understand how that even works. But if all of that can happen, what have you accomplished? You have a prokaryotic cell with a mitochondrion. Do you have a eukaryotic cell?
No.
No, you don't. Right? So if you can make all that work, right, and that's a big if. That's like if with a capital I and a capital F. If you could make that work, like you could come up with a really good, suitable mechanism, you still have a prokaryotic cell with a mitochondrion. You don't have a eukaryotic cell. This in no way describes the origins of how you get your original eukaryotic cell. All it does is explain the origin of the mitochondrion, if all that were true. We still have a big problem. It's like if you were to ask the question, like, what is your favorite food? And you answered it brown. Okay, well, I'm pretty sure what you mean is your favorite color is brown. That's good to know. But that in no way answered the original question. Right? See, we still have a huge problem. Huge problem. Okay, and again, remember that's a big if you could accomplish doing those things. Yeah, David.
Are these problems like they're shown in the theories? Or?
Yes, so much so that the original proponent of this, her name is, I think she's still alive, Lynn Margulis. This idea was proposed in the 1980s, and it was mocked for like 5 years because of all the issues. But then after like 5 years, somebody was like, man, we really don't have any better explanation, and it kind of gained pretty wide acceptance. But again, it doesn't in any way explain the origins of eukaryotic life. Even if it were true, all it does is explain where the mitochondrion came from. Still don't know where eukaryotes came from. And as you know from what we talked about last time, we have no idea, even if you could make a eukaryotic cell, how on earth you could take that and generate all the different forms of eukaryotes. No idea. So maybe it happened a bunch of times. Maybe we actually generated eukaryotic cells several different times that then diversified. Now you're starting to sound a lot more like like special creations, right? All right. Well, it is 12:38 on Friday, and because it's Friday and I want you all to have a wonderful weekend, we'll end class right now at 12:38. Actually, it has nothing to do with me wanting you to have a great weekend. It has everything to do with we finished the chapter. So, but I do want you to have a great weekend. That's just not why class. Wow, that was. I'm sorry if that came off as weird.